Alright everybody, we're going to try doing this online video. So this is the second half of chapter 9. We talked about DNA previously, now we're going to talk about RNA. So RNA is single-stranded compared to DNA that is double-stranded and exists in the double helix. It consists of long, unbranched chains. We're going to see the same phosphodiester bonds. We will just see that it's a different sugar. This is the deoxyribose as opposed to the regular ribose. Pyrimidine bases are uracil and cytosine. So uracil is the addition to the RNA instead of the thymine. So this is the way that information travels in the cell. It's stored in the DNA. In order to get the information from the DNA into a protein, it has to be transcribed. And so the transcription codes the information in the DNA into something called mRNA. So it's the go-between. And then the mRNA is translated by tRNAs, so multiple steps of RNA interactions here, and we'll see some others as well, into making the protein. So information for protein is stored in the DNA, but the DNA has to write it as RNA before it can be expressed as a protein. So we're going to talk about the role that the mRNA and that the tRNA play, as well as some of the catalytic RNAs that are in the cell. And you see one of them actually right here, the ribosome. Oh man. Okay, so first the role of mRNA in transcription. So transcription is the process of getting the information from the DNA into the RNA. So DNA has to be unwound from the double helix so that certain enzymes can get in there and actually start to make the RNA. And the one that does this in prokaryotes, and in eukaryotes, is our RNA polymerase. So we have an RNA polymerase. Now in prokaryotes, it's a little simpler of a system. Let's say that RNA polymerase is going across this gene region, traveling this way, and as it goes, it's going to transcribe some mRNA sections, and this section right here, between these two lines, would be the gene A section, the gene B section, the gene C section. These are each going to make a peptide. Now, as this mRNA is being formed, these little ribosomes, which are actually associations of protein and RNA, do the actual translating into the protein. And in prokaryotes, these little ribosomes can all start lining up on the mRNA as soon as it forms. Now, this doesn't quite happen in eukaryotes. Eukaryotes have two reasons why this doesn't happen. One, all of this process happens in the nucleus for us. This is all just floating around in the cytosol for the prokaryotes. We do have some free-floating ribosomes, but we also have some membrane-bound ones, and those are on the outside of the nuclear envelope. So inside the nucleus, there aren't ribosomes already making proteins. Also, we have an additional part of our genome that actually doesn't turn into protein. It's called these introns here. So we call exons as the coding region and introns as this sort of non-coding region. We don't know what it does. But one of the things that has to happen is that the RNA or that the introns need to be cut out. They need to be cleaved before our mRNA is ready. So there's a processing that has to happen. So we have a few different types of RNA. We have transfer RNA or tRNA. This is what's going to bring the amino acids into the ribosome to help with the growing peptide. We have ribosomal RNA. This is the catalytic part of the RNA of the ribosome that's going to make the protein. Messenger RNA is that go-between between the DNA and the protein. We have small nuclear RNAs, small interfering RNAs, microRNAs, and long non-coding RNAs that we'll talk about towards the end. So tRNAs are single-stranded. You can see a secondary structure right here. These are hydrogen bonds. You see this weird little clover leaf kind of layout. This is where it kind of gets its T name from. Intra-chain hydrogen bonding happens. and the amino acids get added to this 3' end. There's a little modification that'll happen here. A sequence will get added to help the tRNA sort of find that amino acid. I think it's mentioned in your book. Ribosomal RNA, this is the one that actually goes and makes the protein. So this binds to the mRNA. This ribosomal RNA is usually associated with proteins. You can see here 60 to 65% of the total weight of the ribosome. The protein portion is about 35 to 40%. It's different in eukaryotes than prokaryotes, different from one organism to the next. This is kind of how we see the differences, or how we compare a lot of the different lineages. We look at the ribosomal RNA. Two subunits, there's a large subunit and a small subunit, and they're usually given the term 16S or 30S or something S. And the S just refers to their sedimentation rate. It has to do with how large they are, and it's a coefficient of their sedimentation. This doesn't mean that, I mean, for the most part, as the proteins get larger or as the ribosomal units get larger, S increases, but that doesn't, it's not like proportional exactly, because shape does matter also. Here's a little bit of that process, ultracentrifugation to get these ribosomal units to separate. And here's a prokaryotic ribosome. So here you can see we get this guy out from the initial homogenation and lysis of the cell, followed by fractionation. So now you've got the ribosomal units, and these you can subject to the increased centrifugation. Now this one in prokaryotes is a 70S, and it breaks into two pieces, a 30S and a 50S. Now these don't add up to 70 because they have not. That's not what this is. That's not what these numbers are. These are just showing those two different sedimentation rates, and they again are just different from each other. We can actually get these to break apart further, and you see that they have a bunch of different proteins associated with them, and some of them even go into smaller and smaller subunits. Ours is an 80S and not a 70S, so it's a little bit different, but our book does have a little details of, or shows some pictures of ours. Again, most of them act the same way though. So there's multiple subunits, protein parts, and RNA parts. Now the messenger RNA is what the ribosome goes and binds to, and we're going to go explore most of these in detail later. The messenger RNA is formed as a precursor to the protein. It's formed directly from the DNA. It's written in RNA. It's very short-lived. These things are typically made so that the protein can be made, and then once you've got protein, the mRNA gets degraded so that those nucleotides could be recycled to make another mRNA, because often when cells are replicating or dividing, right, they need to double all of their proteins, double all of their membrane proteins, everything. So they need to make lots of mRNA. The complementary strand of mRNA is synthesized along one strand of unwound DNA. Let's see if we have a picture of that. I guess not, so make sure to look in your book and see a picture. We're going to go over the details, like I said, in a different chapter, but here, the starting from the 3' end is something that we'll focus on a little later. I keep doing that. Small nuclear RNAs, these are actually found more recently. They're small in that they have 1 to 200 nucleotides, so that's where the name comes from. They have some function with the processing of the pre-mRNA, so the cutting out of the introns, and to getting it into its final form. So they have some role in that. RNA interference, this is when we use RNA to actually go and interfere with the production of protein. So RNAs can be made as a complement to mRNA sequences. This will go and complementary bind to an mRNA, and while they're bound, that mRNA can't be translated. And so it's a way of sort of interfering with the production of a protein. There are small interfering RNAs that have been found that do this job. MicroRNAs, also similar to the small interfering RNAs, bind to mRNA and prevent its translation. Small interfering RNAs have an additional feature. They cleave the RNA. So this has been used in the treatment of Ebola virus. They use these small interfering RNAs to go and actually mess up the mRNAs that the Ebola virus needs to replicate. So that's pretty neat. CRISPR's pretty cool. You can read the book about some of the details on how it works. Long story short, using CRISPR and the CRISPR-Cas system, you can isolate specific areas of the DNA that you're after, and then you can go and cut them out. And then there are other technologies that let you go and put back in certain genes that you want. And so this is the tool, become the tool for gene editing, finding a gene, cutting it out, and then replacing it, or just cutting it out and seeing what that gene did. This is a hijacked system. Bacteria normally were able to do this to fight viruses, and we were able to kind of hijack that system and use it to do our own sort of genomic investigations. Long non-coding RNAs. These have also been found floating around in the cells. Lots of long non-coding RNAs. We don't really know exactly what they do. We just know that if we take them out, or if we knock them out, with interfering RNA, that stuff usually doesn't work right. I don't remember if the book says specifically that the offspring are inviable. I think there's studied in mice, but they are necessary. We just don't know exactly what they are doing. This is just a graphic, I guess, kind of showing us some stuff here. So RNA coming in here and finding particular regions of the DNA and then cutting it. And then I'm not really sure what exactly this picture is all showing us. Some various aspects of transcription and translation. Just how sort of interfering RNAs work too. So this small interfering RNA has complexed with the mRNA, preventing a ribosome from being able to come and do anything. And then this one also cleaves it. And then this thing here, an mRNA, we've got a ribosome, protein that's growing, and a microRNA that's come in here and messed up the whole process here. This ribosome can't keep going that way because of this microRNA on the mRNA. Anyways, so there's the rest of Chapter 9. I'll be posting a video for Chapter 10 soon. Go ahead and take a look at the quiz that I think I put up there for Chapter 9 on Canvas. I should be posting some questions for you guys to take a look at as well. I opened a discussion for Chapter 20. I'm sorry, for Chapter 9. I'm making other videos for Chapter 9. So go ahead and if you have questions, post them there. And if you don't get an answer there, email me. We'll see what we can do. Stay healthy out there.